Welcome everyone. Today we're going to discuss clinical trials, how you can find out about them, how to find out if you might be eligible to take part in one, and we'll also look at genetic testing results we receive. How can we understand, for example, what the results mean? For those of you who don't know me, I'm Patrick Walker. I'm a journalist and an Alport patient. Alport UK are running this workshop for the Alport Syndrome Alliance, which is a global network of patients, clinicians, laboratory scientists, and pharma companies who are trying to advance treatments and knowledge. As with all Alport workshops, we'd love you to briefly introduce yourself. So just type in the chat a few words about what country you're from, whether it's raining, how early it is, that sort of thing. To help us learn a bit more about clinical trials and understand genetic tests, we have three amazing clinicians on today's panel. Firstly, Danny Gale. Danny directs RADAR, the UK Rare Renal Disease Registry, a UK Kidney Association initiative designed to pull together information from patients with certain rare kidney diseases. He also runs the Family Kidney Clinic at the Royal Free Hospital in London, specialising in the care of families with inherited kidney problems, including Alport syndrome. Francis Flinter is a consultant whose research mapping the gene for Alport syndrome led to her specializing in clinical genetics, with a particular interest in inherited kidney diseases. As a clinical geneticist, Francis has met hundreds of patients with Alport syndrome to discuss the implications of their family histories and the option for genetic testing. And finally, helping me with the questioning today is Judy Savage. She is currently professor in the Department of Medicine at Melbourne Health. Her laboratory works on inherited renal disease and curates a database for the second commonest cause of inherited renal failure, Alport syndrome. Helping us today, Dan and Alice. They will run the production of the webinar today, so do message them in chat if you have any problems. If you're new to the format, or indeed new to Zoom somehow, we usually have some housekeeping to make sure you're getting the most out of our presentations. Danny will present for about 10 minutes, after which we will have a short video from an Alport patient, Ryan Wicks, who's talking about his experiences taking part in a clinical trial. Then we'll follow that immediately with Francis's presentation before opening the floor to questions. If you want to ask a question during the presentations, please use the Q&A function in Zoom. If you wish your question to be anonymous, just click on the anonymous button, and we'll try and get through as many of those as we can today. To share any message in the chat function with everyone who's attending, please make sure that you click send to all panelists and attendees above the chat. That will mean that everyone can see it, not just the panelists. And one thing we do want you to know about, we are not able to talk today about anyone's individual genetic tests or results. So please talk to your doctor or indeed email Alport UK if you have any queries about your individual genetic tests or kidney results. If you have any difficulties using any of the features today, hit us up using the chat function and Alice will be on the case. So I think it's just about time to kick off. Danny, would you like to share your screen and start the presentation?
Hello. Hi. I hope. Thank you for the introduction, Patrick. I hope you can see my screen.
Yes, we can see everything. Yeah, working right.
So thanks very much for the invitation to talk this evening. And I'm going to talk a little bit about clinical trials more in general for the first part of this evening. And I'm going to talk initially about where we are with Alport syndrome, and it'll be obvious to I think most of us here that there's no approved treatment yet for Alport syndrome. Why is this? Well, there's probably several reasons. One is that it's a rare disease. Another is that it affects children, which makes clinical trials harder to do. And the third issue is that the progression to severe kidney damage typically takes place over years rather than weeks or days, meaning for reasons that will hopefully become clear, that clinical trials are somewhat difficult in this population. But this will hopefully change in the future, and there are efforts underway to develop treatments for Alport syndrome. And I'm going to talk more about the testing of new treatment strategies this evening. So if you look up clinicaltrials.gov, which is an international website run by the NIH, National Institute of Health in America, it lists 4 clinical trials, that is interventional studies, which include people with Alport syndrome, that have run recently in the UK. The first of these is ELX02, which is just opening at the moment, and we'll hear a little bit more about that because this is the only one that's currently recruiting in the UK. And that targets people with Alport syndrome who have a particular type of genetic change, which Francis will discuss in more detail. There's been recent trials of lademirsen and bardoxolone methyl. These trials are both now closed. And the information from these, I think, is unlikely to lead to them being introduced as approved therapies. And the final trial is of atrasentan, which is a more broad kidney protective agent that includes Alport syndrome, but also other kidney diseases. So may or may not give us some really important information for treatment of people with Alport syndrome. However, despite the rather small number of drugs on the left side of this slide, we know that there are plenty more drugs in the pipeline, and it may be that the similar slide put up next year will look rather different. So why is it that we need to do clinical trials? Well, when determining what is fact and what is not fact, there are a number of approaches that can be used. There's assertion by trusted individuals or received wisdom. There's empirical evidence to test a hypothesis, for instance, the effect of an intervention. And there's extrapolation from facts that have been established by evidence. But for this last approach, you really need to understand the system well enough to be sure. And that works well for engineering applications like aircraft and cars and most of our daily life. But in clinical medicine, the human body is so complicated that the only way that we really know the effect of a treatment is through clinical trials. And it's important to note that the reason for doing clinical trials is not just to find out whether interventions work, but it's also to identify what harms, if any, they cause. And that allows a balance to be, or a judgment to be made about the balance of harm and benefit. So where did clinical trials originate? Well, the first generally accepted clinical trial was by James Lind in 1747, when he was studying British sailors who were suffering from scurvy. And his approach to this was to take 12 patients all suffering with scurvy and then divide them up and expose them to various supplements or foods, including cider, various types of acid, and a spicy paste. And what he saw by doing this objective experiment was that the group that was given oranges and lemons did very well, and they all got better from their scurvy, whereas the other groups did not. And this really illustrates the point that you don't know what the optimum treatment is, so do a fair test to try and identify which agent, if any, is providing benefit. But there's also another issue, which is it ethical to do trials? And this dilemma was really explored very well in the Dallas Buyers Club, which is a film that came out probably 10 or 15 years ago now. And the basic premise is if there is a new treatment, then surely all patients should have access to it and should have access as fast as possible so that they can get the benefit. But the key point is that this assumes that the new treatment is A, effective, i.e. that it works, and B, that it's safe and it doesn't cause adverse effects. Sometimes there can be laboratory or circumstantial or anecdotal data that suggests an agent will be effective, but in fact, when it's actually used in the real world, adverse consequences can predominate. And the only way to really get certainty about this, or greater certainty about this, is through clinical trials. But this conflicting need between the individual's need for treatment as soon as possible and the needs of other and future patients to determine whether a treatment is safe is really what the medical regulators have to navigate, and that's really the ecosystem in which clinical trials sit. So some examples of where clinical trials have really changed our perception. The first one is bone marrow transplantation for solid cancers. So chemotherapies were developed in the 1970s and 1980s, and they were highly effective, but the limitation on the dose was the effect on bone marrow. So the idea is that you harvest some bone marrow, you store it, you give what would be a lethal dose of chemotherapy to kill the cancer, and then you give the undamaged bone marrow back. However, undergoing this procedure is extremely unpleasant and hazardous, but it seemed like a good idea. The logic is clear. And doctors, patients and hospitals lobbied very hard in the 1990s to make this available. And in fact, there's one case where an insurer in America was sued for not funding this procedure for a patient. And in fact, tens of thousands of patients received these bone marrow transplants for solid organ cancers. But when robust trials were finally done in the 2000s, it was very clear that the outcomes were very much worse. This doesn't apply to bone marrow transplants for bone marrow diseases like leukemia and lymphoma. So nowadays, we don't do bone marrow transplants for solid organ tumors. So that would be like lung cancer or liver cancer or kidney cancer, because we now know that the harms of going through the procedure are greater than the benefits. Another example that's perhaps closer to home for people interested in kidney diseases, erythropoietin. So EPO is a hormone that's made by the kidney and increases the red blood cell count. In kidney failure, lack of EPO, because the kidneys don't work, causes anemia, where the blood count goes very low. And anemia is clearly associated with illness and death, and is a barrier to exercise and activities of daily living. Artificial EPO injections were introduced in the 1980s, and they were very clearly effective at increasing the red cell count. And it was assumed at that time that increasing the blood count to something approaching normal would be the most effective treatment approach. But it wasn't until, again, the mid to late 2000s, when large-scale clinical trials revealed that patients targeted to a normal hemoglobin, a normal blood count, actually did quite significantly worse than patients targeted to a slightly low blood count level. And this caused quite a lot of puzzlement when the Choir study was published, but then the similar results were seen in a second study, and it's now very clear that EPO is used to target a slightly low blood count rather than a normal blood count. So these are two examples where a clinical trial has given us results that are somewhat surprising and have improved the quality of care that is available by not exposing patients to risks. So clinical trials really underpin the idea of evidence-based medicine, and the aim of clinical trials is to define the balance between benefit and harm. And this applies to new drugs, but also repurposing existing drugs for new indications. To get a license, that is approval to market a drug, an application needs to show clear evidence of benefit outweighing the risks, and those risks must be fairly ascertained. And this requires clinical trials that ideally show the benefit on a hard clinical endpoint. That is an outcome that is clearly important to patients. So death would be a hard clinical endpoint, occurrence of kidney failure would be one. Surrogate markers of efficacy such as amount of protein in the urine or the changes in blood count, are less persuasive, but in some circumstances they can lead to approval of a drug. So how are drugs developed? Well, drug development, broadly speaking, breaks down into a preclinical phase and a clinical phase. In the preclinical phase, a model system is developed to study the disease, and a way of intervening on the disease is invented, which might be a small molecule, it might be a gene, it might be any one of a number of approaches. And if the lead chemical or treatment potential therapy is safe and effective in the model system, the next stage is to find out how the body handles the drug, and if the drug has toxic effects, firstly in animals, and secondly in early clinical trials. So the key features of a clinical trial is that participants should make an informed, voluntary decision to participate, and are always free to change their mind. And this is really absolutely critical to doing trials that are ethical and also likely to be scientifically valid. The trial must in itself be able to yield useful results. It's got to be large enough and long enough to see a difference between patients getting or participants getting the therapy and not getting the therapy, and it also needs to be large enough to detect evidence of toxicity. It's also got to be rigorously designed and implemented so that the results are not skewed, and that's not a trivial undertaking, particularly with rare diseases where there might be multiple sites, multiple hospitals participating in the trial across the world. Now, a key process in almost all clinical trials is randomization, and that is where participants are randomized to either receiving the study drug or not receiving the study drug, and this counters the effects of unascertained differences between the cohorts starting out. In most trials, there's also a blinding step which prevents bias of reporting of side effects and ensures that the information that is obtained is objective. This whole process produces a lot of paperwork and a lot of regulation and auditing is needed to ensure that the highest standards are adhered to at all the study sites. And this process is really what consumes an awful lot of time and money, and is the major barrier to implementing clinical trials. There are three main phases of clinical trials before a drug is approved. The first is phase 1, which is to find out if the drug is safe in healthy humans, and this is where a small number of healthy volunteers are given 1 or more doses. Secondly, there's a phase 2, which is to find out if it affects the disease in the way that's expected from the laboratory work and all the preclinical studies. And typically this includes small numbers of patients given the drug to determine safety in patients and also allow an estimate of what the therapeutic effect in real people is likely to be. And typically this type of study will use a surrogate endpoint. Finally, if the drug is found to be potentially safe and effective in phase 1 and 2, a larger study is carried out to really quantify the therapeutic effect. That is, find out just how good an intervention it is in terms of affecting the important clinical outcomes, which might be kidney failure, might be death in some circumstances, and also be large enough to ascertain any adverse effect. And those are the phase 1, phase 2, and phase 3 studies that you may hear talked about. Only a very small proportion of phase 1 studies actually go on to lead to a marketable drug. A slightly higher proportion of phase 2 and a significantly higher proportion of phase 3 drugs. Phase 3 trials actually lead to an approved therapy. So once a drug has gone through clinical trials, the next step is for regulators, which is the FDA in America, the MHRA now in this country, and other regulatory authorities elsewhere, have to make that judgment as to whether it is an effective and safe intervention. Then there's a period where the price of the drug is negotiated between the company that makes it and healthcare providers. Then it's marketed to clinicians and given to patients. And then there's a process of post-marketing surveillance, which is where any undetected adverse effects that might be rare but serious can be picked up. And this is a slightly old slide now, but just gives a rough ballpark figure to the amount of time and money that goes into generating an effective new treatment. So typically more than 10 years. Average cost is estimated at $2.6 billion, although I think this number is, there's quite a lot of error in that figure. And this figure also includes the costs of a failed project. And even bringing a drug that's approved for a different indication to patients with a disease which has not previously been approved for is also eye-wateringly expensive. So that's a sort of rough outline of clinical trials. I'm now very pleased and very grateful to be able to introduce Ryan Wicks, who's a patient with Alport syndrome, who's participated in a clinical trial, and he's been generous enough to share some of his experiences in a video, which I think we're going to show next.
It was a 2-year trial. They actually tried to put me on one a year before, and I didn't meet their criteria because it wasn't under 70%, I believe. So the following year, another trial came up that Professor Gal was quite keen for me to go on, and I wasn't apprehensive. They explained it quite well that it's 1 injection a week. The nurse would come to here, she would take blood, and then she would inject the trial drug into the abdomen. They would have a courier waiting to ship the sample off to Belgium, and for a period of time I had quite a bad side effect to it. I did report it to Professor Gal and the team, and they couldn't really tell whether it was due to the drug or anything else. But there were weeks where within about an hour, and I called it like having a bad trip, that as it comes into the system, I would either be sweating, I'd be shivering, I'd have to go and sit in the car sometimes, or I'd have to go and sit down over there. It was after 12 months, and I understand that after 12 months it became an open trial. So for the first 12 months it was a closed trial, so nobody knew if you were getting the placebo, if I was getting placebo, who was getting what. But after 12 months, everybody got the real thing. So it could have been, I'm not an expert, but I was having the placebo, and then when I did have the initial real drug, I got them side effects. Prior to the trial, it was quite fluctuant, but I think that was also a time when they were trying to get the right medication. But since the trial, it seems to have been very stable at around the 68% mark. It had a bit of a dip, but now it went back up, but it's been very stable for the last 12 months. For myself, it seems it could have worked well because it stabled my function. And should there be another trial, I'll always be happy to participate, because if it does help me, it can help someone else.
Thank you, Ryan, and thank you to Danny for a very accessible presentation. We are open to questions, so please use the Q&A function if you would like to ask one. We're going to move on to Francis's presentation now, but just as an addendum to Danny's presentation, if you want to help understanding what mutation you have and whether you're eligible for a clinical trial, we have a number to do that. You need to call +44 207 472 6491. Now, Dan's just put that number in chat if you would like to call it. Again, that number will help you understand what mutation you have and whether you might be eligible for a trial. They'll take your details and they may call back quite late, perhaps in the new year, but please keep chasing them if you don't hear anything. Right, that's it. Francis, over to you.
Thank you very much, and good evening, everybody, or good morning to those in Australia. Can you see my screen?
It's working great on our end, Francis.
Great, thank you. So I've been asked to talk about how to read a DNA report and how to understand what type of alteration or mutation people might have in one of the Alport genes. And I'm sorry that some of this is inevitably going to be quite technical, but don't worry about the details. The key things that I want people to understand is, sorry, I'm just trying to advance the slides and for some reason they are not going down. Oh, that's frustrating. There we are. There's the key questions that I would like people to think about. So first of all, do you know if any affected person in your family has had genetic testing? If a variant was found in one of the collagen 4 genes, has that variant been classified as significant or not? And sometimes the word you'll hear used is pathogenic. That simply means disease causing. The next question to think about is, have close relatives of someone who's got a known variant in one of the collagen 4 genes been offered testing for the variant that's been identified? And finally, have family members had an opportunity to discuss the implications of their genetic diagnosis and of their genetic test result if they have had a genetic test? So I was asked, first of all, to try and explain what is DNA? It's a term that people often use, but don't necessarily quite know what it is. So DNA is what we inherit from our parents. And it's interesting that across all types of animals, including humans, our DNA is made up of exactly the same language. And it's basically a set of instructions that we inherit that help tell our body how to assemble proteins from amino acids. And it's a very compact language because there are only 4 letters in the DNA language. We've got 26 letters in our alphabet, but there are only 4 letters in the DNA language. And those 4 letters are arranged in words that are always just 3 letters long. So there are 64 different combinations that you can have, and those words code for just 20 amino acids, and the amino acids get strung together to form proteins, which can then assemble to form collagen. So here's a picture of collagen. It's sometimes described as a triple helix because you can see there are 3 chains here, 1 red, 1 blue, and 1 green. And each of these protein chains is made up of a string of amino acids all attached together end to end. Now, we're all different from each other, aren't we? And that's what makes us so interesting. And every single one of us has more than 1 million variants in our DNA, but most of those variants will have no significant effect. And one of the reasons why genetic testing can be quite tricky is that if you sequence somebody's DNA, you'll find a huge number of different variants. But for each variant, you've got to decide, is that variant significant or not? And more specifically, in this case, is this variant possibly something that might explain why a person has kidney disease? So I said that the DNA code is written in just 4 letters. Those letters are known as nucleotides, and the 4 letters are A, C, G, and T. And I've written there what each of those letters stands for. So the genetic code is completely unambiguous. Each 3-letter word codes for just 1 amino acid, or it may code for an instruction to stop stringing together the amino acids. So the amino acids are strung together to form a protein chain, such as collagen. And what happens if there's a variant in the collagen gene, which means that the protein chain isn't made normally? What then could happen in the kidney is that the filter mechanism in the kidney doesn't work properly and it becomes leaky. So on the left-hand side here, I've got a healthy kidney. There's blood going through the vessel there. It hits the filter, and what is excreted out into the urine is just the substances in the body that we want to get rid of. And this healthy kidney is retaining the albumin, which is a type of protein, in the blood there. On the right-hand side, I've got a damaged kidney. So there's an alteration in the collagen gene, which means that the protein isn't made properly, and that makes the filter mechanism leaky. And so some of these albumin particles are leaking through the filter and being excreted in the urine. Now, here's our very efficient 3-letter word dictionary. And the way you read this is you consider what's the first base in the 3-letter word, what's the second base, and what's the third base. So at the top here, we've got TTT, T, 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 and TTT will code for an amino acid called phenylalanine. In fact, TTC also codes for phenylalanine. We only need to be able to code for 20 amino acids, and we've got 64 different triplets. So for some cases, different triplets will code for the same amino acid. Here we've got leucine, which could be coded for by CTT, CTC, CTA, or CTG. But you can see how changing just 1 letter, here if we changed CTT to CCT, instead of coding for leucine, you will code for proline. And there are 3-letter combinations, TAA, TAG, and TGA, which code for stop. And that simply stops the protein assembly. No more amino acids are added to that chain. So here's our 3 stop codons, sometimes known as nonsense or termination codons, because at that point, the protein chain stops being assembled. Now, there are different types of mutations that can occur in the DNA. Some of them are very large. People may have the whole of their Alport gene, their collagen 4 gene, missing. That's known as a deletion. Or there may be a large chunk of extra DNA inserted into the middle of the gene. That's known as an insertion. Or part of the gene may be copied unnecessarily a second time, and that's known as a duplication. And in the very early days of DNA sequencing, we were only able to detect some of the larger mutations. So some of the patients who had their DNA sequenced with a result being found in the very early days are much more likely to have one of these large-scale alterations. But more recently, as genetic sequencing has become much more sophisticated, it's become possible to detect really tiny changes in the DNA, down sometimes to being able to detect just a single letter change or a single base change within the DNA. Now, I'd like you to imagine that you're reading in a language where there are only 3-letter words, and the normal sequence in this bit of the gene is these 5 3-letter words here. The cat ate the rat. It's a normal sequence of 3-letter words, and each 3-letter word makes sense. If just one letter is changed, if that C were changed to a B, that particular word would be different. Cat has become bat, but the rest of the sentence reads okay. The bat ate the rat. And that single letter substitution that just affects one word is known as a missense mutation. One letter has been substituted for another. If you start the sentence, but then you get what we call a stop codon, a stop message, then none of the rest of the sentence is read. So that's a nonsense mutation. Everything comes to a halt. You're just about to start off making your protein assembly and you get a stop message, and so everything stops. Now, another phrase that people may sometimes come across is that they're told that they have a mutation that causes a frame shift. And in this particular example, it's an insertion, just one extra letter stuck in here, a J, then shifts all the other letters along one place. So if you look carefully, bat ate the rat is still there, but because everything has shifted, it's been called a frame shift, and that message is now out of frame. So the whole of the rest of the message reads as nonsense. Now, just to show you in a different way how different types of mutations can occur, a substitution is when just one letter, a T in this case, changes to a C. So that's a T to C substitution. An insertion is where between the C and the T, we've had another C inserted. And you can see that that's going to have the effect of throwing the whole of the rest of the sequence out of frame, because the 3-letter triplets that you would normally see are all going to be misread. And here we have a deletion. Instead of going CTG, the T has been deleted, and that message goes from C to G. And once again, that causes a frame shift. And the last example I'm going to give you is an inversion. So here, TGCA swivels round to become ACGT. Now, the two words that I was particularly asked to explain to you, the difference between missense mutations and nonsense mutations. So missense mutations are point mutations. That's usually a substitution of one letter for another, which will just change a single amino acid. But even though only one amino acid is affected, the consequences can be serious. And it's interesting that all patients who have sickle cell anemia have the identical missense mutation. But missense mutations can also be benign, they can be harmless. They may change an amino acid in a protein, but because the amino acid which is changed actually is changed for another one which is fairly similar, the protein may still be able to work reasonably well. So here is another example. We're looking at a sequence of DNA where the same letters keep being repeated, CAT, 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 and that codes for histidine. But at this point here, instead of CAT, we get swapped to CCT. And that means instead of that particular triplet coding for histidine, this particular triplet, CCT, codes for proline. So that's the wrong amino acid. And that may mean that even though the rest of the protein chain is read and the rest of the protein is made, the protein might not function properly. Now, nonsense mutations are a particular type of missense mutation where the amino acid change results in the production of a stop codon, TAG, TAA, or TGA. And these stop codons do not code for an amino acid, but they basically are a signal that says stop making protein. And what happens if you hit a stop codon prematurely is that usually you then make a truncated protein, a shortened protein, which is usually not functional. So here's an example of creating a nonsense mutation. We've got another set of triplet repeats, CAG, 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 all coding for glutamine, but a single change here from CAG to TAG causes a stop, and the whole of the rest of that DNA can no longer be read. The functional effect of a nonsense mutation depends on where it is within the coding sequence. If it's very near the end of the chain, it's near where the original stop codon was, it may have less effect than if it's near the beginning. So as well as identifying what the genetic alteration is, we also have to think about how is the variant going to affect the protein that will be produced. And then after that, how will that change affect kidney function? Now, I'd like to just talk you through finally a few DNA reports so that patients who've got their own DNA reports can have a look at them and see if they can try and understand a bit more of all the very technical language that's there. Now, the first thing to say is that I always check who the DNA report is on. There's check at the top the name, the date of birth, the patient's reference number, their reference number, what specimen was, when it was taken, when it was received by the lab, and so on. Sometimes people bring DNA reports into the clinic and they say, oh, here's my uncle's DNA report, but then you look at the name on it and actually the name at the top is of a different relative. And it's obviously very important to know whose results you're looking at. A DNA report will then usually say, why has the test been done? And in this patient, it's a diagnostic test for Alport syndrome. So obviously they've got symptoms that their doctor thinks might indicate an underlying diagnosis of Alport syndrome. And the report will then tell you what the lab's done. And in this case, they've looked for sequence variants, deletions and duplications in this group of genes. So the type 4 collagen genes, collagen 4A3, collagen 4A4, collagen 4A5 and collagen 4A6. So those are the 4 different types of collagen genes which can be affected in patients with Alport syndrome. And it also tells you which sequencing technology they used. And finally, there's a comment that when they did find a variant, that was then confirmed by testing for that variant in a different way using an even more precise sequencing method. And in this patient, the result shows that in the COL4A5 gene, which is the gene for X-linked Alport syndrome, at position 1117, a C has changed to a T. And then the report goes on to tell you what effect that has. Well, the effect that it has is that the arginine amino acid at position 373 has been changed to a termination codon or a stop codon. And that is interpreted as being pathogenic, it's disease causing. So in summary, it says the COL4A5, that's the gene for X-linked Alport syndrome, that sequence variant results in a premature termination codon and is predicted to be pathogenic. And so they conclude that this result confirms the clinical diagnosis of Alport syndrome. Now on the back or at the bottom of the page, there'll be a lot more text, which it has to be on the report because it will tell those who need to go into the detail exactly how the testing was done, what proportion of mutations that particular testing strategy is likely to pick up and so on. And it also tells you how the individual amino acids have been numbered because as things are updated and we learn more about the sequence of each gene, the point at which we stop and start counting tends to change over the years. So people will need to know which form was used when reporting that particular alteration. Now, this is a different DNA report. This is a patient who was again referred for a diagnostic test to see whether there was an underlying explanation for the blood in their urine and so on. It tells us that they've been screened for these 3 type 4 collagen genes, so not for COL4A6 in this case, and also another gene called NPHS2. And in this patient, 2 different alterations have been found in the COL4 genes, this time in the alpha-3 chains of the type 4 collagen gene. And in the same way, we can look and see that in the first mutation, there is just a deletion of a single letter there. And the effect that that has is, instead of there being a glycine, which was what we would expect to see in the normal sequence, there is a frame shift. So the rest of the message can't be read properly. And the second alteration there, position 4486, is that a C has changed to a T, and the effect that that's had is that an arginine is changed to a termination. And just a subtle difference in the language there, that second mutation is described as a likely pathogenic sequence. So there's not quite the same degree of confidence that this alteration is significant as there was about that one. And so if we read summary at the bottom, it talks about the first sequence variant is predicted to produce an out of frame transcript and result in premature termination, very likely to be pathogenic. And the second sequence variant results in a premature termination codon and is likely to be pathogenic. And with the conclusion at the bottom, they say that assuming that these sequence variants are present in trans, that means assuming that one of them is present on the chromosome that was inherited from the mother and the other present on the chromosome that was inherited from the father, then this result is consistent with a clinical diagnosis of autosomal recessive Alport syndrome. And finally, just to look at what happens if a relative is referred for testing. So in this case, somebody's been referred to be tested for the familial sequence variant. The patient is the mother of an affected male who's already known to have this particular alteration in COL4A5, a change from a C to a T at position 4165. So the patient herself has intermittent hematuria, and it says she's an obligate carrier because her brother is also affected. So a woman who has an affected brother and an affected son is what we call an obligate carrier. She must be a carrier. But the test is done just to confirm, as expected, that she does have that single alteration in one copy of her COL4A5 gene. And the summary here is that the individual is a carrier of the familial pathogenic sequence variant and therefore a carrier of X-linked Alport syndrome. And some reports will also have a comment at the bottom that's intended for the referring clinician to say carriers of Alport syndrome should be offered lifelong monitoring of their blood pressure and urine to test for proteinuria with a low threshold for being given a drug known as an ACE inhibitor. Now, in some reports, you may see the use of a 5-point classification system that looks at how significant or otherwise variants are. So class 5 variant is definitely pathogenic. It means we've seen it in other families. It's been well reported. We have confidence that this is definitely the cause of the Alport syndrome. A class 4 variant is likely to be pathogenic. It may have been reported a few times and there's an emerging body of evidence to suggest it's likely to be pathogenic, but not quite as confident as a class 5 variant. Class 1 is definitely benign, it's well recognized, it's frequently seen in normal populations. We know it has no effect. Class 2 is probably benign. Again, it's something we're seeing quite frequently. The most difficult class is class 3, variants of uncertain significance. And this means we have no evidence that it's likely to be disease causing, but no evidence that it's likely to be benign either. And so class 3 variants are usually reported mainly because it is possible that in the future, once we learn more, we may be able to reclassify that variant as either being significant or not. So why might interpretation change over time? Well, let's say we had a variant found in one affected member of a family and we thought it might be significant, but then we find that other affected members in the same family don't have that variant. That tells us that that variant cannot be the cause of their disease. The amount of information in population databases and disease databases, such as the one that Judy curates, is increasing all the time. And that's very helpful for labs who are trying to classify new variants that they find maybe ones that they haven't seen before. And of course, although we tend to oversimplify things by focusing just on the genes that we have screened, we have to remember that there are lots of other genes in which alterations can be found, and they can have a modifying effect on people's clinical features. So just to remind you once again, these are the key questions to think about. if you are a patient, have affected people in my family had genetic testing? If there's a variant, is it classified as significant? Have close relatives been offered testing? And have family members had the opportunity to discuss the implications of their genetic diagnosis? And I will stop there. Thank you.
Thanks very much, Frances. As always, a fantastic explanation of genetics, understanding your results and the conclusions I think that people should draw from them. I want to open the floor to questions now, both from me and Judy and everyone watching at home. So if you are watching us at home, please go to the Q&A button at the bottom of the screen and pop in your question. In the meantime, I'm going to hog the floor a little bit with a few of my own. Danny, I know you're a doctor, clinician, but you're also a dad. If you had the decision between putting maybe your child into a clinical trial with a potentially interesting drug that may help solve or treat their disease, is that a decision you would make? How would you think about the risks of entering a trial?
Yeah, I mean, taking on the risk for oneself is one thing, but exposing your child to such an intervention is a very different proposition. And I think really it would depend on the nature of the disease that they've got and what the likely spectrum of outcomes that they are going to have with what we now know about their condition. And then I'd look very carefully at what the quality of evidence was for the intervention, so how likely it was to actually help, and also what the known risks were, and also how much information there is to sort of narrow down the potential risk. But it is, I mean, this is on my first slide, I think I put up that diseases that affect children are much more difficult to do clinical trials in because of this issue, that it is a significant barrier. So I think it would depend on the child and the outlook for the child and what is known about the drug.
And just on that point, why trials are so difficult to run in diseases that affect kids. I think Alport syndrome has also been particularly challenging to find drugs that treat it particularly well. Could you talk about some of the difficulties that explain why Alport syndrome struggled with having lots of clinical trials in the past?
Well, I mean, it's a combination of factors. Being a rare disease instantly makes being rare makes a disease less amenable, partly because the potential rewards to a commercial organization for developing a treatment are smaller, but also, and I think more importantly, doing a clinical trial requires recruiting enough patients to be able to make a determination of safety and efficacy. And if the number of patients with the disease is very small, then there's going to be problems getting enough people into the trial in a time frame that's manageable to be able to actually successfully run the trial. And I think that it's that consideration, it's the rarity that's been the major barrier to doing clinical trials. There's a separate issue which is about development of potential therapeutics, and that really reflects the biology of the basement membrane and how little we've understood about it and how much more we now understand about that biology. So the more we know, the more possibilities there are to go down that journey of developing a treatment. But yeah, Alport's, like many rare diseases, has really suffered from not having large numbers of potential therapies or large clinical trials. And also the slowness means that you have to do a trial that's long enough to see an effect. And of course, if you could only recruit small numbers of patients, then it has to be even longer. So again, if you can recruit 10,000 people to a trial, then a small difference over a short time can be really powerful. But in Alport syndrome, that's just never going to be the case.
We heard from our patient Ryan in that wonderful video by Sam Clark, who's an Alport patient himself, talking about some of the side effects or potential side effects that he might have experienced during the trial. What what safety checks are put in place during the procedure of a trial to ensure that patients are kept safe and informed and that side effects don't become problems?
Yeah, I mean, it's so a clinical trial isn't just of this nature, isn't just giving a medication and then coming back in 6 months. As he mentioned in the video, he was having blood tests every time just before he had each dose of the drug. So we were monitoring very closely the various parameters, looking for ill effects, and of course discussing with him at every stage whether he wanted to continue or not. And we looked very hard to find evidence of sort of medical harm. And in the absence of that evidence, then it's a matter of balancing the benefits of successfully completing the trial and the inconvenience or disbenefits of whatever side effects are encountered, and that is an individual choice. That's why consent is absolutely critical to the whole process. You've really people have got to understand what they're likely to see, and then they need to know that at any point, if it's too much, they can stop taking the trial medication.
And as always, I think the common thread between all of your answers there has been just educate yourself, make sure you're as aware as possible about the trials, potential side effects, about places you can go if you have issues. I have an anonymous question that I just want to ask you, Danny, about a trial being done by ELOX. I'm not sure if you're aware. They want to know how many of the community might be eligible, what you know about it.
Yeah, so the ELOX trial is the only one that I'm aware of that's recruiting in this country, and that's being run at the Royal Free and Great Ormond Street Hospital, as well as sites in Australia. And that targets a very small subset of people with Alport syndrome. So they have to have nonsense mutations, which Francis explained beautifully. And that's around probably 5 to 10% of people with Alport syndrome. But the catch is that those types of mutations in general tend to progress rather more rapidly than some other types of mutations. So it's targeting a particularly young group who have this rare type of mutation in a rare disease, and that's really one of the main challenges for this type of study is identifying patients who fulfil the eligibility criteria.
So we have a tiny subset of an extremely rare disease. So good luck to the guys there. Thank you so much, Danny. Judy, I'm conscious I want to get to Francis, but would you like to have your go at interrogating Danny while everyone else asks their own
couple of questions. Thanks, Patrick. There's a couple of questions in the chat, and I might just start out by answering them and then asking the panel some questions. So somebody said that she has been diagnosed with Alport syndrome but has no other family members. And so does that mean she has a genetic mutation or not? If you have a mutation, it's a genetic mutation. You have a genetic disease. About 1 in 6 people with Alport syndrome, with X-linked Alport syndrome, have nobody else in the family with it beforehand, but they will in subsequent generations. So, but the only way to be certain is actually for family members to have genetic testing. There's all kinds of tricks involved in this. So I think it would be worthwhile for other family members, for example, brothers and sisters and children, to also have testing. There's another question here, and I wanted to lead into asking Francis about this, about who gets genetic testing. What happens if someone hasn't had genetic testing but actually wants to know? What happens in the UK, Francis?
So in the UK, there's a national system to try and make it equitable for everybody for whom genetic testing could be helpful. And I know Danny's very involved in overseeing all of this. So genetic testing is not something that your GP can request, unlike some other blood tests. Genetic testing to look for alterations in the Alport genes can be requested by your kidney doctor or by a clinical geneticist. So if you want to find out more about whether or not there is a genetic alteration in your family, you need to ask your GP to refer you to the local genetics clinic, or next time you're in the kidney clinic, ask the doctors there and explain to them why you think genetic testing would be helpful for you. I know sometimes the response may be, well, we know what diagnosis you've got. We don't need to do a genetic test to confirm that you've got Alport syndrome, because we can see you've got kidney disease and we know you have some hearing loss. But of course, genetic testing is also helpful for other members of the family, because once you know the variant, you can test the relatives to see if they've inherited the same variant. And it can also help the individuals themselves to know which type of Alports they've got, because that will affect what the chances are of their children inheriting the same condition. And so particularly if people are getting to the age that they might in the future be thinking about having children, that could be information that's helpful for them. So people need to be prepared to have that sort of conversation with the person with whom they're discussing genetic testing.
So I think in jurisdictions where the testing is not available freely, there's often still ways and means of getting it. For example, being in a clinical study, often that study will offer genetic testing before you start in it. There's also some research laboratories throughout the world who will do genetic testing too. So it's worthwhile speaking to your kidney doctor and just finding out if these things are available.
And I think Danny wanted to add a comment there as well.
Yeah, sorry, yeah. I'd just say that in the UK, the system has changed a little bit just in the last couple of years with this new genomic medicine service. And it's now, and this wasn't the case previously, but it's now centrally funded by the NHS to do genetic testing in anybody with a diagnosis of Alport syndrome, or indeed with familial hematuria would be an indication for testing. So there may be people who were diagnosed years ago when genetic testing wasn't available, and then there would be no prompt to their clinician to initiate genetic testing, but it would now be available on the NHS.
Francis, can I also ask you, does everyone get a copy of their genetic test result? And if they don't, are they able to?
So people can always ask for a copy. They won't necessarily automatically get them. I used to ask my patients whether they wanted one or not, and some would say yes, please, and others would say no, thank you. But yes, every patient has an absolute right to have a copy of their own genetic test result. And if they decide to share that with their relatives, then obviously it helps their relatives when they go and request genetic testing, because it's telling the other doctors which variant to look for.
On that, I just think we see on Facebook a lot that people try and compare genetic results when they they have access to that page. Could you talk a little bit about your advice on how accurate it is to try and compare results between one person and another?
It's tricky. I find genetic reports quite challenging to read. I have to read them very, very carefully, and it's a specialty I've been working in for more than 30 years. So I certainly wouldn't do it as a coffee time chat. You need to be able to see the whole report. You need to know which lab the testing was done in. If it was done in a research lab, you can't use that information clinically. It needs to be validated. It needs to be replicated in an accredited service laboratory. And I think it's because almost every family with Alports will have a different mutation. You can't really compare them. They are all different. And I think it's better to have a conversation about the interpretation of your result, either with your geneticist or with your kidney doctor, or with the case of the new study that's about to be launched. You can ring the telephone number that was put in the chat earlier and talk to somebody who does understand these things, but they will only be able to comment on the report when they have read the whole report, including the footnotes. You can't just extract out one line and compare that, because you've then only got part of the story. You do need the whole jigsaw puzzle in front of you before you can comment.
Thanks. I'm sorry, Julie. I just wanted to get in with that. Don't compare genetic test results.
Danny, can I ask you, why are there so many trials in Alport syndrome? And they seem to be looking at different things. Why is that so? And which would be better to go in?
So there are a number of, I mean, the kidney is a complicated organ. The human body is a complicated system, and there are many different ways in which the change in the type 4 collagen chain leads to kidney damage and kidney failure, and indeed hearing loss and other manifestations. So there are lots of different ways in which potentially that system can be intervened on to improve things for patients. So for instance, the atrasentan trial looks at an inhibitor of both the sort of ACE inhibitor type mechanism, but also a hormone called endothelin. And there's evidence that that's protective across a wide range of different kidney diseases. And it's quite likely that it's going to provide some benefit to people with Alport syndrome, but the magnitude of that benefit is unlikely to be transformative. So going into a trial of a drug like that, you will be looking, the investigators will presumably be aiming to recruit very large numbers, hundreds or even thousands of people, and looking for relatively small differences in outcome, but they'll have lots of people to see evidence of safety. In more Alport focused, particularly rare Alport subsets like the ELOC study, they'll be looking to intervene very close to the actual cause of disease, that is the genetic cause of disease, and they'll be looking to see a big difference in effect, so a big effect. And if they don't see that, then the trial won't show a difference between people getting and not getting the drug. So there are lots of different ways that one can approach diseases. There's a sort of general therapy that protects the kidney that might actually provide a lot of benefit, particularly for people with more slowly progressive disease. And there's other approaches which are more tailored to Alport syndrome, but are of necessity going to have much fewer patients involved. So those are two examples of reasons.
So one of the questions was, why don't these trials actually address the genetic mutation? But you've intimated that some of the trials are starting to do this directly.
Yes, so the ELOC study, for instance, the reason it only recruits people with stop codons is because the way the drug works is it encourages the cell to read through the stop codon to essentially get round the genetic problem. There's also work going on, and there's been preclinical studies of putting the gene, the normal copy of the gene, to try and rescue the kidneys, and those I think are at stages of development, but they're not yet in clinical trials. They've not proven to be effective enough in model systems and safe enough to warrant going into humans yet. But that, of course, that might change in the future.
Okay. You also mentioned that often the study is an active agent compared with a placebo. So people might be having a lot of tests, a lot of inconvenience for, say, a year, but not being treated with the active agent. So are most of those people given the active agent afterwards? Is that a common feature of clinical trials?
So it depends on the trial and the type of trial and type of agent. But for rare diseases particularly, I'd say it is fairly common that if there's evidence of efficacy after the trial period is over, then the company is going to need to have more safety data. So they're going to need to have more people on the drug to gather more information about benefits and risks to go to the regulators to get it approved. So I'd say that it's fairly common in rare diseases studies for there to be what's called an open label extension phase of the study, where everybody who was in the study gets the drug. Now, clearly, if the drug didn't provide benefit or significant benefit, then being on as an open label is also not of benefit. But in the instances where the drug is eventually licensed and is shown to be effective, then those people, even if they missed the active drug in the placebo part of the trial, they can potentially get many years of open label extension drug that they would not have got if they'd waited for it to become available. But every study is different, and the pros and cons of open label extensions sort of vary with the drug.
I just wanted to ask Francis now. Francis, the genetic test result is actually incredibly complicated. Why is that? Is it possible to have just a single line summary, or what should people be looking for in the result?
So I suppose the single line summary is either there is a pathogenic variant detected, or there is no pathogenic variant detected. That's the simplicity thing. It's yes or no, isn't it? The reason it's so complicated is firstly because virtually every family with Alport's has a different mutation, and there's all sorts of different ways that the DNA can be altered. And scientists around the world have spent a huge amount of time liaising with each other in order to come up with a common language to describe the alterations that they find. And because the sequencing can now be done with such incredible depth and accuracy, they can be very precise in terms of describing exactly what the alteration is. Where does the error occur? What is the error? Is it simply the change of a single letter, or is it a bigger change than that? Is there a chunk of DNA missing? In which case, where does the missing bit start and stop? So the language is complicated because it has to describe hundreds and hundreds of different types of alterations. I think it's been simplified as far as it can be. And the language that we use now both describes exactly what the alteration is and then what effect that will have on the protein chain that the gene should be coding for.
Can I ask you another question, Frances? And that is, if a person had, for example, a great uncle who had Alport syndrome, and now their child is suspected of having it and is undergoing genetic testing, and maybe they don't speak to their great uncle and certainly don't know what mutation it is in the family, how can they access it, or how confidential is that information? And how easy is it for the lab, say, to access the mutation that the great uncle had, or a distant family member?
So every family is different, aren't they? If there are known to be other affected individuals and they haven't been tested, or they're completely uncontactable, then the starting point may have to be the child who has symptoms, and the lab will have to do a complete screen of all the Alport genes and try and find the alteration there. If from the family tree you know that the child's mother is what we call an obligate carrier, then it may be that rather than taking a blood sample from the child, you could take a blood sample from the mother, and you'll be able to identify the familial mutation that way. If family members are not talking to each other, but people happen to know that the uncle has had genetic testing and they know his name and date of birth, then providing that information to a laboratory may enable the laboratory to look up the uncle's result and know which alteration to test for without necessarily needing to disclose any information about the uncle to the family. They can simply then tell the family whether or not that particular alteration has been identified in the person who has requested genetic testing. Genetic testing is highly confidential, and one of the jobs of clinical geneticists is to make sure that we both keep information confidential, but also help families to share information with other relatives as and when is appropriate. And sometimes relatives don't want to talk to each other, but they're happy for us as clinicians to write to their relative's GP and suggest that the GP's patient should be tested for Alport's without necessarily even disclosing who the relative is, who is affected, if the family doesn't want us to do that. So there are all sorts of ways we can try and share information if it's appropriate to do so without breaking any confidences. But it is quite tricky, and we have to sit down with people and spend some time discussing what are the issues, what are the concerns, how can we try and help, how can we support the sharing of information without sharing, other things that families would prefer to keep confidential and private.
Could I just finish up by asking, what are some of the risks of genetic testing to both Danny and Frances? And yesterday I spoke to somebody in the clinic. The father has had a genetic diagnosis, and I know his son does not want to be tested because he's very anxious about the outcome. But what are the risks of genetic testing, and what about that anxiety? You must have seen it many times in your patients, and then a reluctance to be tested. So what are the risks of genetic testing?
Well, genetic testing is never compulsory. People have a choice. And sometimes just allowing people time to think about it and to think through the issues means that they may come back in a few weeks or a few months' time and say, right now, I'm ready to be tested. People need to know what the outcome of genetic testing could be. It may confirm that they do have a particular genetic diagnosis, or it may exclude that genetic diagnosis, or the results may come back as uninterpretable, and actually we may be no further on. And that is always a challenge. We always encourage people who are considering genetic testing to be aware of the fact that the information generated by their result could be of relevance and value to their relatives. And so they need to start thinking about how they will share that information with their relatives if that's something that they feel able to do. And very often with a supportive geneticist or genetic counsellor or nephrologist, if it's the nephrologist having that conversation with them, given support, given time, very often people can get to a stage where they decide that the benefits of hopefully obtaining useful genetic information and being supported when they consider the implications of those results and what it means for other members of the family, give them enough confidence to decide that, yes, they will go ahead. It's not a decision that needs to be rushed, unlike many other things in medicine. There's always time to think these things through and to find somebody to talk to who understands what the issues are and can think about that individual's own particular situation.
Thanks, Frances. Danny, did you have something to add?
I don't really have much to add to that excellent answer that Frances just gave, but I would say that there is a difference between a genetic test done in somebody who is known to have the disease and you're finding out more about that disease, compared with somebody who has just found out they might be at risk of the disease. And then it's a question of really predicting for them what they're at risk of in the future, and that has got huge pros and cons. It might, testing someone and finding that they're unaffected obviously is of enormous benefit to them, but testing someone, finding that they've got the condition might have a big impact. It might change their plans, it might change their plans about having children, it might change their career plans, it might change the way they think about themselves. So it depends on how old they are when they're being tested and what sort of where they are mentally at that time. So it is worth thinking carefully before, as Frances said, you do normally have time with these sorts of decisions to think carefully. So careful thought is obviously recommended.
Thank you, Jan. That's a really important point. And I would just say that for predictive testing, so somebody's currently healthy but wants to know if they've inherited a susceptibility, we very rarely test people the first time we meet them. They usually need more than one appointment to discuss it before they go ahead and have the test. Whereas diagnostic testing for somebody who's clearly got symptoms and you're trying to find an explanation for it, usually the decision to go ahead and do that test is more straightforward, and it's more likely to be something that's done more quickly.
I would have to say that in my experience, genetic testing has often revealed surprises, whether it's the mode of inheritance, that it's autosomal recessive rather than X-linked, or whether it's actually not Alport syndrome, but another X-linked disease. So until we have the genetic testing, it's really hard, in my mind, to be certain about the diagnosis. And I think, although we're talking about clinical trials here, I think it's important to remember that there are treatments currently that help delay the onset of kidney failure by years and years. And it is worthwhile having blood pressure monitored closely and stopping smoking and making sure you don't get overweight, etc. So there's a lot of things that can be done, including medication, that will delay the onset of kidney failure. I'm going to hand back to Patrick. Thank you.
Thank you so much, Judy. We did get a question in the chat that I want to finish with. Frances, is there an online resource that people can use with that has some of the content in your talk?
Well, first of all, I think this talk's been recorded, hasn't it? So
it is, yes. It's going up on our YouTube as soon as possible.
For people. Yes, probably best if I look up the links and send them to you, but Health Education England has got some excellent online teaching packages for people who want to learn more about genetics and genetic mutations and so on at various different levels, depending how much depth people want to go. into. So I will look up those links for you, and then perhaps they can be posted on the
Thanks. Could I just interrupt, Patrick? There's also a question here. What is the best source of patients for patients or clinicians to find out about clinical trials? And that's for Danny. So how can people find out about what trials are available currently or what might be coming?
Yeah, so there is an international resource, or American resource that I mentioned called clinicaltrials.gov. And that's a sort of massive database that may be quite difficult to filter through, and you've got to pay very close attention to which trials are actually open. But what that will do is give you an indication of which hospital sites are open for a given trial. In the UK, we've got the Rare Renal Disease Registry, or RADAR, which all patients with Alport syndrome are eligible to enter. And RADAR at the moment is being set up so that patients who, as far as the data held by RADAR can ascertain, seem to be eligible for a clinical trial, will be notified automatically, and they give consent for that notification when they join RADAR. So that's a mechanism where we hope will raise awareness of clinical trials going on. There's also a RADAR Alport group website that in due course I'm hoping will have more clinical trials to allow people to sort of hunt down clinical trials for specific diseases. And of course, one's own kidney doctor will also might know of clinical trials that you might be eligible for. So it is certainly worth asking about. And of course, Alport UK is a pretty good resource, and they've got a fairly clear view of what's going on in this area in the UK.
I was going to say I'm happy to plug our own channels when it comes to learning more about Alport syndrome, but I'm going to mention in the outro an email, if I can scroll to it, an email that you can hit up for to learn a little bit more about either your own genetic testing results or genetic results in general. Thank you all three for a really, really useful discussion. I think this is the most engaged we've seen people in a while in workshops, and that's a tribute to really how well everyone explained it and how good the questions were. So thank you so much all for your inputs. And as I said, this talk will be recorded and stuck on YouTube, hopefully within the week. So if you want to go back over anything that has been discussed, you can do that. My takeaways really from this one are to ensure that you're informed and to communicate with your physician as much as you can about what choice might be best for you, either in terms of clinical trials or getting genetically tested. I also want to thank particularly Francis for outlining the mechanics around genetic testing. I think we get one of our most common questions is why genetic testing takes ages, and you've helped explain that a little bit. It's important, I think, that as always, we finish our workshop with a word cloud, which many of you I'm sure will be familiar with. Dan has put a link in the chat, and I'm going to demand that everyone go there right now and put a word in that describes how you're feeling having watched our workshop today. I'm going to put mine in. I'm not going to tell people what I put. Informed, I think, definitely something that I felt very strongly. Optimistic as well, yes. And as Danny said, I think we're only really at the start of what trials could be available, but that all really hinges on us participating in them and being informed about what trials are going on. Intrigued, yeah, me too. Me too. Enjoyable. I don't think we've had that one before. I feel like these. I think everyone's just delighted to have two presentations that are just really, really accessible. So thank you so much, both of you. Or maybe that's just me. Very informative. Okay, fantastic. So thanks everyone for coming today, and please do let us know if you have any topics for research or research that you'd like us to talk about in our future workshops. Before we go, I want to plug the Alport UK online Christmas get-together. That's on Tuesday, the 13th of September at 7 p.m. UK time. It's just an excuse really for us to chat over a cuppa. It's not going to be about clinical trials or anything, but it's really, really informal.
You said September. We don't
Did
wait.
I say September? Oh, thank you, Francis. 13th of December.
Fine.
December. Oh my God, I'm going mental. Please don't forget your Christmas jumper. It's a date for your diaries, the 7 p.m. Tuesday, the 13th of December. We're planning our next Alport social, which will take place in Cardiff at some point in the spring. So also look out for that. Huge thank you to Danny, Ryan, Francis, Judy, Dan, and Alice for helping put the workshop together. Thank you to the patients who have contributed their thoughts and questions, and of course to Ryan, who shared his experience in the video. Finally, we appreciate Kidney Research UK for their sponsorship. They help fund our Zoom subscription and keep the workshops going. If you have any feedback about the session or want to be added to our mailing list, email workshops@alportsyndromealliance.org. Alice will paste the email address into the chat area right now in case you missed it. Thank you, Dan. Patients can join the Alport Warriors Facebook group to chat with other people with Alport syndrome or who are carers of people with Alport syndrome, and anyone can hit us up on Twitter and Instagram, both at Alport UK. As I said, we'll have a talk of this, a recording of this talk. We're going to get through this, one more sentence to go. We will have a recording of this talk on YouTube in a few days' time. Otherwise, thank you all so much for watching, and have a lovely rest of your evening.